Hello, thanks for joining us. Welcome to today's webinar. My name is Tim Finnegan, and I'm just going to help facilitate the webinar today entitled Drowning in Data, Monitoring Harmful Algal Blooms. I want to kick things off by introducing our first presenter. Dr. Stephanie Smith is the product manager for YSI's environmental monitoring systems. She's responsible for the management of multi-parameter water quality instruments and for the development of new monitoring technologies for surface waters. Dr. Smith holds a doctorate in microbiology from The Ohio State University, and she's held various positions in academia and industry, including running her own business that offered products and services related to harmful algal blooms. Stephanie is going to actually cover the first half of this webinar with her presentation on algae, and then we're going to turn it over to Chuck Springer. Chuck's been managing water data for well over a decade, previously working to manage a large state agency's water database. He has a master's degree in environmental science, and he works here at Aquatic Informatics to help clients design data management solutions that really help turn some of their raw data into actionable information. Chuck's going to pick up at the second half of this webinar after a poll where he's going to demonstrate how to use a spreadsheet tool to manipulate some pH and chlorophyll data. And then he'll also show a commercial tool to manage and follow those same processes, just with a different workflow. Just a quick bit of orientation before I hand things off to Stephanie. We're going to try and keep the webinar to around 45 minutes to save some time for questions at the end. And as I mentioned, it's generally going to be in two parts, a presentation by Stephanie and a demonstration by Chuck at the latter half. At any point during the webinar, I encourage you to ask questions. We'll do our best to respond to those questions both in real time and save some time at the end to answer those. I apologize in advance if we're not able to get to everyone's. We actually have a very record number of attendees, so it is going to be difficult to get through everything. But if we don't, feel free to type that question in, and we'll do our best to get back to you within a day with a follow-up. And I will just mention we are going to follow this this webinar with an email to the recording. We have a link to a video that we're going to reference, an Excel sheet that'll be shared. So there'll be a number of resources that you'll get in an email after this webinar if you're attending live. So with that, I'm actually going to change presenter and let Stephanie take it away. Stephanie, you want to take it from here?
Yes, I do. And it looks like I need to change slides here. There we go. All right, thank you very much for that introduction, Tim. And I want to thank all of you that are online with us. I really appreciate your interest in this topic. As you are interested in it, I wanted to remind you that this is a follow-up. It's part 2. The part 1 was a webinar we did at the end of July, and we're going to send you that information. You could also probably even just search this title on YouTube if you want to look for that. But in that first webinar, I covered a lot of the principles about harmful algal blooms using this infographic. And what we tried to demonstrate and communicate was that there are a number of environmental factors that impact a harmful algal bloom. Some you're very familiar with, like nutrient runoff, weather conditions. But the other thing we made a point of was that the algal bloom itself impacts the environment in turn. And that's why a multi-parameter monitoring approach for a lot of these different things is really your best bet when it comes to monitoring for harmful algal blooms. So we talked about how some conditions really impact the growth of algae, but then the algae also impacts some of those conditions. So if we think, for example, of carbon dioxide, there's atmospheric CO2, but there's also dissolved gas. And as the algae are consuming that dissolved CO2, that has an impact on pH, and pH also impacts the growth of algae. We see a lot of changes in dissolved oxygen as the bloom cycle, as we call it, occurs. So this is why a multi-parameter monitoring approach approach is really, really valuable. And of course, one of those parameters that people monitor most often when they're interested in algae blooms is chlorophyll. Now, in my first webinar, I talked about both chlorophyll and phycocyanin. And chlorophyll is the pigment that you don't just find in blue-green algae, but actually you find it in all algae, including seaweeds, eukaryotic algae, blue-green algae, and everything on up to higher plants. And of course, that's because all of these organisms are performing photosynthesis, where they're converting the energy from sunlight into cellular fixed carbon. So the molecule itself, chlorophyll, I've shown here, not because I necessarily expected you to recognize it, but if there are any biochemists in the audience, you might recognize the potential here with all these double bonds for this to be a fluorescent molecule. And in fact it is. And we often have monitored for chlorophyll for decades, in fact, not just recently with multi-parameter sondes, but for many, many decades. Chlorophyll has been monitored as an indicator of the size of the algal population, and the general rule of thumb is that the more chlorophyll you have, the more algae you have, and that is all algae, not just the blue-green algae. So it's a very useful monitoring tool, and in the interest of time for today's webinar, we're going to focus on chlorophyll as a monitoring tool, but I do want to remind you that there are other pigments involved, and I'll remind you of that throughout the talk. So of course, the way I prefer to monitor for chlorophyll and to monitor harmful algal blooms in general is to use what we call a multi-parameter sonde. And here I'm showing you YSI's EXO TAL sonde, which has a payload of a number of sensors that you can use to monitor conductivity and temperature, chlorophyll and phycocyanin, pH, dissolved oxygen. But the sensor of most interest today is what we call the total algae, or TAL sensor, which allows us to monitor those photosynthetic pigments. So just a little bit of a reminder to understand the principles for how this sensor works. The total algae sensors and some of our other sensors use the principle of fluorescence. And a fluorescent molecule can absorb energy of one wavelength and emit energy of a longer and lower energy, a longer wavelength and lower energy. This is a little reminder of how that works in the context of the electromagnetic spectrum that you may have seen somewhere in your education. But the relevance here today is that individual photosynthetic pigments have unique absorption and emission spectra, meaning they absorb at unique wavelengths of light and they emit unique wavelengths of light. And here's an example of absorbance spectra for preparations of chlorophyll and phycocyanin that I prepped in the lab years ago. And this is again showing that electromagnetic spectrum. And chlorophyll absorbs many, many wavelengths of light, but it doesn't absorb well in that sort of yellow-green region, which is why it looks green. Likewise, phycocyanin has a nice absorbance peak here that starts in the greens and goes on through the red-orange, but it doesn't absorb in those shorter blue wavelengths, which is why it appears blue. Note that on these graphs we're not showing you the fluorescence, we're just showing you the absorbance. The way we take advantage of that is we've designed a couple of sensors. The one that gets used the most often is called the total algae phycocyanin, or PC sensor. That is what we call a dual channel sensor, and it emits wavelengths of light that excite both chlorophyll and phycocyanin. This sensor is most useful for monitoring blue-green algae in a freshwater environment. And not just blue-green algae, but all other algae because of the chlorophyll component of the sensor. Now, if you're interested in the blue-green algae in a marine environment, we might advise you to use the total algae phycoerythrin sensor because those algae, the blue-green algae in a marine environment, have a pretty good load of this pigment, phycoerythrin, which helps you distinguish it from other algae that might also have some phycocyanin. That's an interesting phenomenon that happens in the marine environment. But really, either of these sensors can be used if your main interest is monitoring chlorophyll. So the general principle of how the sensors work is that the sensor has two primary LEDs that emit light. There is an orange, a yellow-orange emission of 590 nanometers, which, if you recall, phycocyanin can absorb light in that range. So it gets, we say, excited by that light, and then it will emit light of a longer wavelength and a lower energy. It just so happens that that light emitted by phycocyanin can be absorbed in the cell by chlorophyll. In fact, that's one of the ways that photosynthesis works, is by what we call fluorescent resonance energy transfer between these pigments. So that's very convenient because we get a nice little boost of chlorophyll absorbing that energy that comes off of phycocyanin. Now, in addition to that, the sensor sends out a blue light that also directly excites chlorophyll molecules. So it's the excitation of chlorophyll from these two sources, the phycocyanin emitted light and the blue light from the sensor, and then the chlorophyll will emit light that includes the wavelength of 685 nanometers that is detected internally by the sensor. So by alternating the excitations from the sensor, we can actually subtractively determine the concentration of phycocyanin versus total chlorophyll by the difference we get when we use these different wavelengths of light to excite the pigments. The advantage of doing it this way is that you get a super sensitive pigment or sensor, excuse me, with really fast response times and high resolution. And we've also shown very clearly that it's less prone to interferences in the water. So here are examples of the specifications for the sensors for the different pigments and phycocyanin and phycoerythrin and chlorophyll. And here's the resolution that you get. But let's turn our attention to these units that we talk about these pigments in. For the total algae sensors, the units that we use are RFUs and micrograms per liter of chlorophyll. And I want to talk about what that means. Now, I want to go back to something that comes up a lot, and that is a unit called CFU per mil. So a colony forming unit is something that is determined in a laboratory where a scientist will grow algae, or this could be an environmental sample, and you make dilutions of it, and then you spread a measured volume of that dilution onto a petri dish. And when you've diluted it enough, you'll get a very nice number of discrete colonies that you can count, and then use that to back calculate what the concentration of algae were in that original sample. This is classical microbiology, and colony forming units are a very standard unit of measure for bacteria, including a lot of cyanobacteria. But some inherent assumptions in that are that the cells you're plating have to be alive, which of course isn't always going to be the case in an environmental sample. And here's a big one, they have to be able to grow in laboratory medium. It actually ends up that less than 1% of microbes in the environment are even able to do that. And one cell forms one colony if you get good separation when you spread them onto the plate. Some cells kind of clump up together. So if we look on the right here, this is an organism called Shigella that a friend of mine was working with a couple of years ago. And you can see that on a laboratory medium, Shigella forms really gorgeous, discrete colonies that are very, very easy to count. If you look on the left, this is a cyanobacterium that many of you will have heard of called Microcystis aeruginosa. It doesn't spread well at all. It's very hard to get good discrete colonies of Microcystis aeruginosa, and it really depends on the medium you use. So it's hard to get a good CFU. And for this reason, there can be a lot of variability in CFUs as determined by one technician versus another. Another unit that you will often see is called cells per milliliter. That's different than CFUs per milliliter because this is derived from typically microscopy. This is what Microcystis aeruginosa, one of our favorite blue-green algae again, looks like under one type of microscope. It forms what are called micro colonies. And what that means is the individual cells tend to cluster together. That makes it really, really hard to get a good count of cells per milliliter. And there can be a lot of variability from one analyst to another in how they are able to get cell per mill counts. Now, something that adds a little confusion to our world is that cells per mill is often used synonymously with CFU per mill, but they are not the same unit of measure, and we are all guilty of this. I've done it before too, but you really have to look at whether that's what people mean when they are using one of these units, so you have to follow up on that. There are new technologies now, FlowCam as an example, some different flow cytometry technologies that do give better consistency in obtaining cell per mill numbers, but they are very expensive and they're really not practical for monitoring purposes. So those are two very, very common units used for counting algae. The total algae sensor is using different units. It's using what we call RFUs and micrograms per liter. I prefer parts per billion of pigment. An RFU is actually a scaled measurement of the output of the sensor. So every individual sensor has a range of output that it's capable of delivering, and we simply calibrate it using a secondary standard, in this case rhodamine, to adjust that scale. RFUs is the default unit for the total algae sensor, and frankly it's the unit I prefer. The reason I prefer it is because you can normalize your whole fleet of sensors to this unit, and it also seems to deliver the most consistent results. It's less dependent on the types of algae you have in an environment than some of the other units we've been talking about. If you must have CFU or cells per mill, you really should build your own correlation with RFU. And that's because of some of the things we've already talked about, how different algae cluster differently. They have different levels of pigments. It really can be variable from organism to organism and environment to environment. But here's a really nice example. You know, it's 10 years old, but it shows very well how RFUs, you know, the range on the sensor, there's a bloom in this situation where you get sort of a peak on RFUs of phycocyanin in this case, and it's really only consuming about 3% of the total sensor's range. So that gives you a really powerful range for monitoring. And what it also means is you can get good sensitivity and very, very early detection using a unit like RFU. Now, parts per billion of pigment is also a very popular unit, and it is one that we offer with the total algae sensors. It was derived using an EPA standard method where we would collect and extract the chlorophyll and measure that in the laboratory using both the sensor and a benchtop fluorometer to sort of ground truth the sensor measurements of RFUs. So this worked pretty nicely for the blue-green algae we were working with in the lab, but you should check at least once how well this correlation works for your samples if you intend to use this parameter. We find that PPB forms a pretty tight correlation with RFUs under most conditions, but we do have some folks that observe it behaves a little differently for them, and it really is dependent on their environment and the types of algae they encounter the most often in that environment. So let's kind of pull some of this together, how you can use the sensors with other monitoring tools. This is a graphic that we used in the last webinar, and we talked a lot about all of these different things. I'm going to talk to you briefly about monitoring satellites that are very useful now for what we do, what we call forecasting. And I'm going to show you how that aligns with a fixed buoy in Lake Erie that is equipped with a multi-parameter sonde and that is using telemetry to communicate to another type of satellite so that you can recover your data. So let's look at an example. This is called the Lake Erie HAB Forecasting Program. You can find this online courtesy of NASA using something called the MODIS satellite. And as you might expect, the more red algae you have. And it's important to understand that the satellite is seeing the surface of the water and is able to do a spectral analysis to help it detect chlorophyll on the surface of the water. So what this means is the more red you get, the more algae are on the surface of that water. And if we look at these three sites, we have a site that really doesn't have a lot of algae at all, a site that appears to have quite a bit, and then a really hot site near Little Cedar Point. And we have gotten pH data off of fixed buoys that were at those sites. But let's look at what else those buoy data are telling us. So at each of these three sites, one of our partner companies called Limnotech has a sonde attached to a fixed buoy, and that has a telemetry system talking to one of those telemetry satellites so the data get delivered to their desktop. That sonde is delivering pH data, and what you may notice is that as the algae density appears to increase, so does the pH. And this is one of those examples of how the algae are impacting the environment because they're consuming that dissolved CO2. The other thing that's increasing is the percentage of dissolved oxygen. And this increases in these heavy density areas because there's very, very active photosynthesis going on. And if you look back at that earlier slide where I showed you the equation for photosynthesis, oxygen is one of the products of photosynthesis. So in the very active throes of an algae bloom, you often see increased dissolved oxygen. Now here are the RFUs of chlorophyll and phycocyanin, the other pigment, for these sensors, and this is what is measured by the sonde. So what's apparent is that there's definitely a lot of chlorophyll. There's definitely chlorophyll, and in the case of phycocyanin at one site, a lot of phycocyanin. But what might surprise you is these numbers don't really look that high. And this is something that you really have to consider if you're going to try to use data like this. So this is an example of where we're trying to ground truth satellite data with sensor data. And the difference might be, for example, that the satellite data is just seeing what is on the surface of the water, whereas the sensor is looking below the surface at a fixed depth. So those densities might be a little bit different of algae at that fixed depth. And this is one example of why you have to really look at a multi-parameter picture to really understand the dynamics of what's going on with an algae bloom. Taking that one step further, I strongly encourage people to collect grab samples at their sites of interest. So you really should use data like these for monitoring and helping you decide if and when you should go out to the field and collect actual samples and make measurements of things like I showed you earlier, colony forming units per mil, cells per mil. You might do a pigment extraction. That is actually really slow and expensive if you had to do that all day, every day, the way a sonde is monitoring for you. So alone, it's not a sufficient tool for monitoring a harmful algal bloom. But when you pair it with monitoring, it really can reduce your long-term costs, and you gain more knowledge and build those correlations over time so that you know how to better use your monitoring data. So Chuck is going to show you how you can combine the power of these monitoring data with data that come in through the laboratory so that you can get the most value out of those data. So to quickly summarize my section of the presentation, the total algae sensors can deliver both RFUs and parts per billion of pigment. I'm a believer in using RFUs, but if you have a preference for using the pigment unit, you should by all means do that and try to develop some idea of how well that correlates with your environmental samples by at least once building a relationship between RFU and either CFU, cells per mil, or parts per billion of pigment. And I find all of these data most useful in the multi-parameter context. So getting the data from multiple tools really gives you the best basis for responding to, monitoring, and managing a harmful algal bloom. But really the question we want to address here is how are you going to manage the actual data? And that is what Chuck is going to talk to you about, and I'm going to hand it over to him so he can show you some examples of how to do that.
Thanks, Stephanie. Before I turn it over to Chuck, I actually wanted to engage the audience for a moment because Chuck's going to be manipulating data in a spreadsheet. And I'm going to do that by kind of launching a poll that you're going to see on the screen. And we'd love to hear some feedback before we break into Chuck's presentation about which types of data, algae data, you may be collecting. So please, if you could choose the ones that are most common to you, we'll kind of look at those results. And those are streaming in. Looks like interesting data changing pretty quickly here. So we'll give those just a few more minutes to click an option on your screen. And it looks like certainly about half the audience so far isn't collecting any through these methods, but then we have a pretty even split between microscope analysis and using a total algae sensor, which fits in nicely. Let me close the poll now and share the results. So you can see here's the breakdown of how folks are currently collecting algae data. And we'll come back to this as we get to the question and answer period. But in the meantime, I'm going to go ahead and give Chuck the presentation so he can start talking about common data challenges and show how to use a spreadsheet tool to start addressing some of those.
Okay, thanks, Tim, and thanks, Stephanie. And so we're going to take a look now at how some of the most common challenges we hear about from folks in the industry are dealt with. And we're going to focus on three challenges that we find are kind of the top things that people are dealing with from day to day. And we're going to look at those, how we can do those using a spreadsheet tool, in this case Microsoft Excel, and then we'll look at those same challenges and how we can address those using a commercial off-the-shelf solution that's really optimized to deal with these very kinds of things, and we'll be looking at Aquarius time series. So our challenges that we'll be looking at today are, first off, managing data from multiple sources. As Stephanie mentioned, we've got continuous sensor data from our sondes and lots of parameters that we're collecting. We've got samples that we're sending off to the lab and we're getting those results back. We've got field data, cleaning regimens and calibration regimens of the sonde sensors themselves, and getting all of this data matched up and lined up and put together can be a bit of a challenge. The second thing we'll be looking at is actually correcting for fouling and sensor calibration drift that can occur in some of these sensors. and how we can address that. And then the third challenge we'll look at is this correlation moving from the RFUs that we're collecting on the blue-green algae and chlorophyll sensors into cell counts and into micrograms per liter. So to kick things off, we're going to take a look in Excel and work through these different challenges. So here we've got some data files for a station on Miami River in southern Ohio. And we've got two files here of data, and these truthfully span multiple deployments of sondes over the course of a little over a year, kind of a project monitoring. And at first glance, they look quite similar, but if we dig in a little deeper and take a look at the headers on each of the first few lines, we start noticing that they're actually pretty different. We've got, from one file to the next, we've got parameters that have moved from one column to another, specific conductance and dissolved oxygen and depth. We've got a cable power parameter in the first file that's absent altogether from the second. So our first task in this first challenge is to get these two aligned so that we can have one continuous file of our year's worth of data, spanning these multiple sonde deployments. And I'm sure this is a challenge that just about everybody out there today has dealt with at some time or another, if not on a daily basis. And it's often a challenge of copying columns from one place to another, or creating a third sheet and moving these things around. And it's a very manual task, and oftentimes what that task looks like is this. We've got maybe two monitors up and multiple spreadsheets, and we're moving things from one place to another. And we're not going to bore you today with spending an hour doing this, but instead we'll focus on the end result here. Once we've got our continuous file of data spanning all our deployments, we've moved these things around, and we've got one data source here for our continuous data now. And this brings us to the next challenge, which is dealing with drift that can occur in these sensors. And as Stephanie mentioned in her presentation, pH is a particularly responsive parameter to algae growth. And it's also one of the more prone to drift. So we're going to, just for time and simplicity, we're going to kind of focus in on pH here a little bit and take a look here. I've cropped our year's worth of data down to just a couple key days worth, and it's a period where we've ended one sonde deployment and started another. And we can see in the highlighted rows here, we've got quite a step change as we took a sonde out that's been deployed over the last month or 2 months, and we put either clean and recalibrate that the sensors of that sonde, or replace it with a freshly calibrated sonde that we brought out from our lab. We see we've got this step change in here, and if I delete this line where we've got a 0 where the sonde wasn't actually in the water, we can see that step change even closer, and we've got quite a misalignment in the pH. And so our next challenge here is to address this step change, and how do we do that using Excel? And the way that this takes shape is actually a fairly complicated procedure that we've got to pull off here. And so in order to do it, I've created a couple new columns in my continuous data file. I've created a new column to house the corrected pH. And I've created a column that I'm calling increment. And this is essentially a row count. And this is counting up the number of time intervals that we have from the beginning of a particular sonde deployment to the end. And here's our step change that we saw on the previous sheet. And here's our corrected pH. And in short, what we're doing here is we're taking this total difference between our drifted pH and our pH from our newly calibrated sensor, and we're applying a correction that's incremented over time. So we have a total of 2,616 time increments. Up here at the beginning of our correction, we're taking 1/2600th of our total correction and applying it, which is effectively a correction of 0. By the time we get down here to the end, we're applying the full difference between our two SONs, and the end result, as we can see here, is a corrected pH trace. And if we compare that to our step change that we had before, now we have incrementally corrected that calibration and or fouling drift out of those data. And realistically, and if we look close at the data, it's not just the pH. We've got the same issue with the dissolved oxygen. We've got it with the turbidity and the specific conductance. So this is a process we're actually going to have to repeat, 3 or 5 times to correct each parameter that we're collecting. That brings us to the next step in this whole process, which is really kind of getting back at challenge number 1 again, which is bringing different sources of data together. And in this case, we've got our lab data. So we're sending off samples, getting extractive analysis of chlorophyll, of blue-green algae. We're getting the results back from the lab. If you're lucky, those come in the form of an electronic data deliverable. And an Excel spreadsheet or a CS file, something that you can go in and actually copy numbers out of and do something with them. If you're less fortunate, and a lot of labs, even in this day and age, still do this, they deliver that data back to you in PDFs or even on paper. And in this case, there's really, unfortunately, no two ways about it. It's the task at hand is keying in all of these results, into a spreadsheet or into some system, and it's opening things up to transcription errors and all of those kind of things with the times and with the values and everything. So once we've got our lab report back, the real task at hand, once we've got the data from the lab in, is matching that up with our continuous data. Really what we're interested in here are just those dates and times in the continuous data where we have corresponding lab data. And when we look closely at this isn't such an easy thing to do either, because oftentimes we're collecting the continuous data once every hour, once every 15 minutes or 30 minutes or what have you, and we're going out periodically and we're taking samples, and the sample date and time isn't exactly the same. We can't automate this and tell Excel, go to our sheet of continuous data and find the value that's at 10:30 on this date, because there is no value at 10:30. It's at a different time. So it's a very manual process of going through and finding the data we're interested in, copying it from one sheet to another. And if we've got a year or 2 years of data, if we're doing this at the end of a project or a study, it can be actually quite a time-consuming thing to pull out the data that's of key interest to us. Once we've done all that, and it's another very manual process, the real task at hand for pulling this data together is, as Stephanie mentioned, doing the correlation between what we're collecting on the deployed instrument, on the continuous instrument, and what we're getting back from our ground truth data, from our lab data or field data. So here we've got an example of a correlation between chlorophyll in RFUs and chlorophyll via our extractive analysis in micrograms per liter, and a resulting regression equation. And we can do that same process to get our blue-green algae RFUs into our cell counts. And so here I'm just going to highlight both of these columns. I'm going to insert a scatter plot, and now on that scatter plot I can tell Excel to add a trend line, and for that trend line we're going to say we want to see what the equation and the R squared are, and now we've got our regression for blue-green algae as well. This equation for this correlation is what we're going to use in our last step, our final challenge, and this is to model our chlorophyll in micrograms per liter from our continuous RFU data. And so this is a matter of taking that regression equation and applying that down through all of our continuous data, and whether we have a month's worth of data or 5 years, we're going to apply this to every cell and get now chlorophyll in micrograms per liter. So now we've got data that is of use to us and that we can use now for further analysis. And those are our 3 challenges. So we looked at managing data from multiple sources, copying data from here to there. We looked at how we can incrementally apply a drift correction to offset our data for calibration and fouling drift, and that Excel spreadsheet, we've sent that out as an attachment here on the webinar. So if you want to take a look at those calculations and dig in and see how we did the various steps, it's all there in the spreadsheet, and you're welcome to download that. And then our challenge number 3, we saw how we can correlate our RFUs into cells per milliliter or micrograms per liter. Now we're going to take a look at these same 3 challenges and how we can use Aquarius time series to address them. And just a quick note about Aquarius time series, it's not a tool like Excel. It is a data management system. It sits on top of a relational database. It's got a web-based user interface, and it's got workflows in it that are optimized for dealing with these very kinds of challenges, and we're going to take a look at those now. So here in Aquarius, I've created our same Miami River location, and I've created some time series, and these are, we can think of these as containers in which we're going to put data from those files that we were looking at. Right now they're empty, there's nothing in them, and we're going to take a look at how we can, one, get the data in, and then work with it here in the system. And so getting the data into the system is, in Aquarius, we're able to take those files and actually drag and drop them into the web interface and ingest the data. And what's going on behind the scenes here, as these are uploading and parsing and coming in, is that we can create configuration templates in Aquarius, and the template is simply a mapping of this column in the file, the data in it goes to this time series in the database. And for, you know, here where we've got 10 or so different parameters, we're mapping each of those 10 columns, and we say what the format is and how the timestamps are oriented. And these configurations are reusable. So what we did was made a configuration for that first SON file, and then we used it as a starting point for the second, and just moved things in the configuration where they needed to be moved, and these are now a reusable thing. And if we get, because we've done that for both of these file types, if we get any more files that match either of those types, we can simply drag and drop those and they come in. And we can actually reuse these templates for other locations. If we have SONs at 10 or 50 or 100 locations in a monitoring network, we can reference these same 2 configurations that we did and bring in data at will anytime we need to. So now that we've got the data in, and we'll actually go and take a look at these data here in a little bit, but first I want to go over to our field visit information. So we've also brought in our discrete data from every time we were on the site, and we did a cleaning regimen, a calibration regimen, we did lab sampling, all of those kind of things. Those data can come into Aquarius as well, and each of those visits are called a field visit in Aquarius. And if we look here at our same visit on May 24th, we've got some basic information about the visit. We've also got our pre and post cleaning readings that we did with our sonde, and as well as an in situ reading after our calibration was done. And the calibration data itself is here, and we can see that we've got pre and post calibration readings in 2 different standards. So we're actually dealing not with just one calibration point here with pH, but we're dealing with 2, and there could just as well be 3 if you're doing a pH 4 standard as well. And because we're taking these readings every time we're on site and we're putting these into Aquarius, over time Aquarius notices that we have a drift issue. We've said there's a difference between our pre and post cleaning, which represents fouling drift, and in this case, it's about 0.2 of a pH unit, or is 0.2 of a pH unit, and it's seeing that we've got differences between our pre and post calibration, and it's alerting us to that magnitude of drift as well, and applying a correction to the continuous data becomes a push button operation. And that correction has now been applied. And now let's go take a look at our pH data and see what we've ended up with. So we've brought in our two data files. The separation between them is somewhere in here in the middle. And we've done this drift correction just on one of our visits, on our first visit here. And I zoomed in a little too far there. And what we see now is the green line represents the raw data. This is a snapshot of the data as it first came into Aquarius, and it remains untouched. We don't modify it, so you can always refer back to that raw data at any time. We see here the colored points are our pre-cleaning, post-cleaning, and then our after calibration. These are all of our in situ readings. And then we see our corrected data represented by the blue line, and here we've matched up the data. It's the exact same kind of correction that we did manually in Excel, this incremental, time-weighted, gradual correction. And we've let Aquarius do all of that heavy lifting for us, and it's applied that correction to the data. So that brings us now to our third challenge, which is we're collecting blue-green algae and chlorophyll in RFUs. What we really want is cells per milliliter or micrograms per liter. We want to take our RFU data and turn it into micrograms per liter. Lucky for us, Aquarius has a way to deal with this as well. So we can take that regression that we've done in Excel, and indeed we would still do that regression in a tool like Excel or like R or whatever your tool of choice is for statistical analysis of data. We're going to take the result and apply an input here of our chlorophyll RFUs at Miami River, and we're going to run them through this equation that we enter in the formula bar, and the result is a continuous record of chlorophyll in micrograms per liter. Now, if we get any new data, if we make any corrections to the source RFU data, drift corrections, or those kinds of things, this will automatically update, and there's nothing further that we need to do. Once we've created the equation, we're good to go. And Aquarius provides REST APIs, web service APIs, that we can actually call directly from Excel. We can request the chlorophyll RFU data and the lab results micrograms per liter, and Aquarius will give us the data just for those lines. Basically, what we saw here, it will give us just those lines where these match up. It will time align them for us, and we can do our regression equation, and there's no need to go and match things up and line them up and all of that. Aquarius does that all for us as well. And the result, and just for fun, let's bring in our pH data as well and overlay that. But the result now is that we have continuous chlorophyll data in RFUs, as you see in the blue, and then here we've overlaid our pH as well, so we can actually bring these data sets together graphically and further analyze what we're seeing. And so, in summary, we saw our three challenges: data from multiple sources, adjusting for calibration and fouling drift, and modeling one unit from another, or even one parameter from another. And we saw in Aquarius, we can create configuration templates and drag and drop the data where it needs to be. We can have Aquarius automatically apply drift corrections for us, and we can set up a calculated data set that will continuously model our micrograms per liter, or our cell counts, from our source RFU data. And
Great. Yeah, thanks, Chuck. Yeah, before we get to some of your questions, we wanted to launch one more poll just to get a little feedback from the audience. If you could take a moment to click one of the options on the screen to talk a little bit about how you may be presently managing your water quality data and your algae data. And some of the early results are coming in. By far, it looks like spreadsheet utilities are the most common ways. And so we hope that by showing how Chuck did that incremental drift correction, you found some value in it. We'll give you a few more seconds to go ahead and input your data. Okay, I'm going to close the poll and share the results. And it looks like, yeah, over 60% of folks are relying on spreadsheet utilities. And that actually is a powerful utility, but sometimes it can be a challenge to scale those results up when you're dealing with a lot of different sites or a lot of different parameters. And certainly the number 2 option was some kind of custom database. Go to the last slide. Now we're going to go ahead and get to some of the questions. And I have 2 that I'm going to pass over to Stephanie. The first one's from Benjamin. He was noticing when you were talking about the Lake Erie data that there was a relationship between algae and pH. Could you just talk through a little bit about how those parameters may be related?
Yeah, I'd love to. Benjamin, thank you for that question. So it's pretty interesting because as CO2 dissolves into water, it forms a lot of different species in water. It can form carbonate, bicarbonate, and carbonic acid primarily, and all of these exist in sort of an equilibrium that really almost buffers the water a little bit. So there's actually a little bit of buffering capacity in the environment. And what happens is the algae start pulling that dissolved CO2 out of that equilibrium. And what that causes to happen is you start losing the balance of some of those more acidic species of CO2 in the water, and therefore you start getting an increase in pH. So the general rule of thumb is that as algae start entering into that really dense population state, they start consuming dissolved CO2 and some of those acidic species of CO2, and you start getting an increase in the pH in the water.
Thanks for that, Stephanie. And then another question for you along those similar lines was from Brian. If a single cell forms one CFU, how come one CFU doesn't necessarily equal 1 cell per mil?
Yeah, that's a great question that every student of microbiology should ask. And it boils down to how it kind of boils down to error and what you're actually looking at under the scope. So remember, in an ideal world, they would align. So let's just put that out there. You're absolutely right. They should be, that should be a 1-to-1 relationship with a perfect slope if you plotted those against each other. In the real world, when you're counting stuff under the scope, realize you can be counting cells that aren't alive. You can be counting cells that aren't going to be able to grow for a number of different reasons. And you can be counting things that you think are cells that aren't actually cells. So all of those things introduce some error. And then the other reason is simply analyst error. One anecdote I'm familiar with was there's been monitoring on a lake out in California. The U.S. Geological Survey has monitored a lake out there for something like 30 years continuously now. And they had a guy that did their cell counts for 25 of those 30 years. And he retired, as I would if I were doing cell counts for that long. And the new analyst that came in was very good, very skilled, very well trained, but all of their data looked different over the next 5 years. And it was simply, there's a real difference in how people look at and count, particularly cyanobacteria, under the scope. So there's a huge variability. Now, there are a handful of people that have actually developed some of their own methods to improve that consistency, but it's one of the reasons that I really wish regulatory agencies would get away from CFU per mil or cells per mil as a parameter of interest, because the variability in it is just huge, and I don't think it's as meaningful as we all wish it would be.
Great. Thanks, Stephanie. That's great answers. And I have a couple here for Chuck. Chuck, one of the questions we had was, are there any capabilities in Aquarius for dealing with uncertainty analysis of the field data?
Good question, Tim. Yeah, with what we showed today, we kept it pretty simple. But we do have, when we were looking at our field visits, and we were looking at our pre-cleaning and post-cleaning readings of pH, we can actually specify, if we know the uncertainty around those field readings, we can specify that as well, and that actually goes into that calculation. So for example, if we have an uncertainty in our readings that exceeds the difference between the pre-cleaning and post-cleaning, Aquarius is going to see that and see that we don't really have a correction to apply because we've got a very high uncertainty that cuts into our fouling drift assessment.
And our next question comes, if you don't have the calibration data or from your field visit, can you manually apply corrections to the data?
Absolutely, another good question. Yeah, so the automatic correction that we saw me apply, that very same correction can be applied here from our data correction tool, and that among many others. So the correction that Aquarius is using behind those auto corrections is this USGS multi-point correction, and the multi-point is that ability really around largely pH, where we've got multiple standards and we're not correcting to a fixed value, but rather a curve of correction values. And we can apply that and any of the others that you see here on the list manually as well.
Thanks for that, Chuck. Stephanie, this is one I'm not sure if you have off the top of your head, but it may have been in your diagram. What wavelength does phycocyanin emit? Do you happen to know that one?
Yeah, let me think for a minute. I'm trying to remember. So it actually emits a range of wavelengths that are in the high 600s. I don't remember what the exact peak is, to be quite honest. I have another slide of that, though, and would be happy to email that out.
Great. And another one was, and this is kind of general, but I think it speaks to some of the challenges we are trying to address with this webinar, is can you talk more about how the YSI sensor information is translated into micrograms per liter? So, yeah, could you just
Sure.
speak to, yeah, go ahead.
Yeah, I went over that in pretty general terms, but so the upshot of it is that when we were developing the sensor, we did some laboratory-based experiments where we actually used a standardized EPA method. We grew cells of a couple of different types in a culture, and we collected those cells on a filter and went through a process like the one I showed in the slide, where basically you collect them on the filter, you perform an extraction, and then you can measure the concentration of chlorophyll either on a spectrophotometer or a fluorometer, actually. And this actually raises a very, very important point. And then those extracts are diluted in water, and that is measured with the sensor. And this raises a couple of important points. One is, bear in mind that the chlorophyll you are measuring in water is actually going to behave a little bit differently than the chlorophyll you measure in an extract, because that extract is formed with acetone or another organic solvent. So that's one thing to be very cognizant of. Another thing to be cognizant of is extracted free chlorophyll is a lot different than chlorophyll that's actually in a viable cell. So oftentimes we talk about in vivo versus in vitro chlorophyll, and that extracted chlorophyll would be in vitro chlorophyll. In the water, where you have live algae, you're looking at in vivo chlorophyll. So those two things can't perfectly align. And this is one of the reasons I say that and then there are other biological layers of complexity on that, like, you know, how does one algae cell versus another type of algae cell compartmentalize the chlorophyll in the cell, what time of day it is. So sometimes cells regulate the concentration of chlorophyll in the cell depending on how much sunlight there is, and all of these things can introduce some variability in those measurements. And that's why you can use parts per billion, and it's still a good indicator for you to monitor trends, but be very cognizant of how a lot of these other factors can have an impact on the correlation of that to the true number of algae. So that's, I hope that kind of gets to an answer, and I'm happy to follow up if there are more questions about that.
And thank you, Stephanie. It does bring us to the end of our hour, so thank you to everyone for attending. When you do close out of the software, you are going to get a brief exit survey with 4 questions, so I'd encourage anyone to give us any feedback through that survey. There are certainly probably another 20 questions we just aren't able to get to in the audience, so if you give us a day, we'll do our best to follow up with those one-on-one through an email. So thank you very much for attending, and have a great day, everyone.